Hello everyone, welcome to Get My Class. So let's solve this question. It says why it is possible to use meristems of certain viruses, virus-infected plants for micropropagation. Option A to produce virus-free plants. Option B to produce SCP, that is single cell protein. Option C to avoid usage of phytochromes, and option D to produce disease-resistant plants. Now let us understand the question. It says why it is possible to use meristems of certain virus infected plants for micropropagation. So what is the reason that even if the plants have been infected by virus, but still the meristems of such plants can be used for micropropagation? Okay, maybe they do have very high yielding capacity, but then because they are infected by virus, so they may get spoiled. But then why is it that their meristems can be utilized for micropropagation? So that is the question. Now, first of all, we have to look for the two important words, that is micropropagation and the other one is meristem. So first, let us talk about micropropagation. So what do you mean by micropropagation? So micropropagation is nothing but production of many number of plants, production of many number of plants, many number of plants by tissue culture. Why? Is it possible to produce number of plants by tissue culture? Yes. Why? Because tissue culture, where you can produce tissues and that means you can culture the tissue, it will give rise to callus, and further you can divide the callus into many bits, and each bit or in each individual cell will give rise to a new plant. Okay. So that is why it is possible to produce many number of plants in the lab condition. Okay. That is by tissue culture. So that is why it is called as micropropagation. So where the sample can be a cell, it can be a tissue, it can be a bud, it can be any part of the plant that can be utilized in the presence of hormones. It will be induced to give rise to new plants. Okay, that's why it is called as micropropagation. Now let us talk about meristems. Meristems, these are nothing but tiny parts of tiny region in the plant which consist of meristematic tissue. They consist of meristematic cells, you can say. Cells, group of cells are called as tissue. So usually they have meristematic tissue in the form of at the tips. Okay, that is you have axillary meristem, axillary meristem. Okay, then you have apical meristem, apical meristem. Okay, apical meristem will be at the tip or the terminal. This can be called as terminal meristem. Axillary are present in the axis of the stems. Okay, these are like tiny bud-like structure, a group of cells which are having meristematic cells, and the capacity, or you can say the potential of meristematic cells, can be described by the term totipotency. Totipotency. So what do you mean by totipotency? That means these have the capacity to give rise to an entire plant. Okay, so each cell can give rise to an entire plant. That is what is the totipotency. Okay, and now here another very important characteristic of this meristem or meristematic cell is that they divide very fast. They divide very fast, that they will not allow the virus to complete its life cycle. So usually virus needs the host, right, to complete its life cycle and to produce more number of particles, viral particles. But here, because they undergo very fast division, the virus is not able to complete its life cycle within a single cell. Okay, so that is why what happens, they will, they try to complete, but by then the cell would have divided. Again, they try to complete, by then the cell would have divided. So by after several divisions, what will happen? The virus will be left out without division, and you get new cells which are free from virus. Okay, so they divide very fast, and so as to give rise to virus-free plants. Okay, so you can produce virus-free plants from these. That is the importance, and that is why it is used for micropropagation. Now let us understand the question. It says why it is possible to use meristems of certain virus-infected plants for micropropagation. Now you understood, even if the meristem is infected by virus, when you culture it, the cell division will be so fast that it will not allow the virus to multiply. Okay. And that's how you get, virus-free cells. And these virus-free cells will be again cultured to give rise to callus, and that will give rise to many plants, and that's how you get virus-free plants. Okay. So option A says to produce virus-free plants. Yes, that is the absolute reason why the meristems are used, even if the plants have been infected by virus. Option B says to produce SCP. No, they don't use meristems for the production of single cell protein. Because single cell protein are used, where unicellular organisms are produced, okay, for the production of single cell protein. The wide usage of phytochromes, not much, because you need to induce root and shoot in these meristems. So you need to provide them good amount of hormones. Okay, hence it is ruled out. So option D says to produce disease-resistant plants. Yes, disease-resistant, that is virus. Sorry, it is not disease-resistant, it is virus-free plant. Okay, remember there is difference between disease-resistant and disease-free. Disease-free in the sense like it was having the disease and now you are getting it disease-free. That is by meristem culture. Okay, virus-free. Okay, not virus-resistant. So these cannot be resistant. These are virus-free. Okay, resistant in the sense the ability to avoid any infection, but these cannot avoid infection in future, though now they are disease free, but they can get affected by disease later. Okay, so option A is the correct answer. Let us conclude the answer. Option A. I hope you understood the explanation. Thank you, everyone.